Perfect. Good morning, everyone. Thank you for joining us today. I am delighted to be hosting today's webinar, The Science and Clinical Impact of Viit Health. I would like to mention to the audience that we will have a Q&A section at the end of the presentation where you will be able to ask questions. During that time, a pop-up will appear on the screen, so feel free to think about your questions while our presenters give their talks. The focus of today's webinar is to hear from experts in their field regarding the science and clinical impact of non-invasive technologies, and especially the impact of Viit Health's device. So I'm very happy and honored to be here with Dr. Adriana Monroy and Dr. Antonio García. Thank you very much for joining us today. Before we begin, I would like to briefly introduce myself. My name is Victoria Merino, and I am today's moderator. I'm a biomedical engineer and chemist with half a decade of experience in neurotechnology and medical device scaling, and I am the Chief Research Officer at Viit Health. At Viit Health, our mission is to make preventative health care painless, fast, and accessible. Our vision is to extend the lives of 1 million people by 10 healthy life years through effortless prevention. Viit Health's device, as you can see in this slide, is able to measure glucose, heart rate, oxygen saturation, and blood pressure. An important fact that I'd like to mention is that the largest cause of death are non-communicable diseases. That means that 43 million people die per year, and 80% of these deaths are actually preventable. In the last year, I'd like to share some of the major milestones we've achieved. We've validated our system in over 3,650 patients. We've reached a technological readiness level of 7, which means that our device has been tested in operational settings. We filed and published US and PCT patents, and we've begun preparations for our US clinical trials and FDA submissions. So I'd like to thank our incredible collaborators and our strategic partners for helping us through this. In terms of the technology, for those of us who are listening to us for the first time, our technology is created and centered around 3 unique techniques: near-infrared spectroscopy, polarimetry, and photoplethysmography. It is the unique blend of these 3 techniques that allows us to obtain an individual spectrum and a spectral footprint for each person. Once the spectral footprint is cleaned and processed, it can be analyzed and used to derive key biomarkers, and we can create a correlated feature spectrum. Dr. Tony is here with us today, and he will talk more about this information. In terms of how we can convert light into clinical insights, we essentially are able to draw raw data from each patient, especially anonymous spectral footprints, and we're able to join all of the information that we get, both sex, age, and other features, and we're able to then compare these features and this data to their heart rate, to their blood pressure, and to their glucose. What this allows us to do is for us to get a comprehensive list of over 50,000 data points. 140 data points are taken per patient, and now that we have more than 3,600 patients, this allows us to train our models and to look at the future of predictive healthcare. At the center of all of this is the health of our patient. What this looks like in terms of validating our technology and where we are today, the following images can give us an understanding of what we've been doing. At Hospital General de Mexico, we're able to validate and compare our technology with current technologies. Viit Health measures in 30 seconds what could take more than 17 minutes. In terms of detection and where we are, in the competitive landscape, we know that there are other multi-metric competitors, such as the Apple Watch, Omron H-Guide, Withings, iHealth, Bodymetrics. But today, we find ourselves very close to the FDA standards that we are here to push through breakthroughs in non-invasive health. What we are able to do, and I think that the importance of this slide is how far we've gotten. In terms of our mean average error, we've reached a mean average error of 1.97 beats per minute in terms of heart rate. In terms of oxygen saturation, we're at a 0.8% difference, achieving a 95.6% accuracy. And then in terms of blood pressure and blood glucose, we're pushing the frontiers to make sure that we're as accurate as possible. But as you can see, in terms of where we are and how close we are to the bullseye of each of these measurements, we have become incredibly close, which means that upcoming with our studies, we currently have 2 active studies and 2 prospective and upcoming studies, we will be able to get the data that is necessary for this to become even more accurate. We've been able to achieve all of this due to the integral members of our team and our key collaborators and partners. So today, I would really like to thank the people who are here with us today, because they're the ones who have allowed us to come to all of these conclusions and to reach these results. So I'm very thankful to be here with such incredible members. I would like to present both Dr. Antonio García and Dr. Adriana Monroy Guzman. Our first speaker is Dr. Antonio García.
Thank you, Victoria. Thank you for the introduction. And welcome to everybody who's joining in. I think you already had a really nice summary of what we're doing at Viit Health. So my job here in the next 9 minutes is to give a little bit more background. And so I think it's always important for me, I guess maybe it's my academic training, is to talk about how we got here from the history of the science and technology. And really, you can trace oximetry, which is the measurement of how much a person has oxygenated in the terms of percentage of oxygen, all the way up to 99+%, to the 1940s. That was the first oximeter, was developed by Millikan and Viervoort and Mathis and others as well, improved it. But it took about into the 70s to get Takuo Aoyogi to really make it practical. And so photoplethysmography was really how this came about. So photoplethysmography is the terminology we give to looking at how the signal varies as your pulse is going through its different phases based on your blood being moved around by the heart. And in that period, the 70s through the 90s, we had a lot of advancements in electronics and optics, making it available and miniaturized. So this led to, in this century, the rise of the wearables, as we heard from Victoria, and also medical grade PPG sensors, where you're able to really get some accuracy in the whole waveform and to be able to understand it in the sense that a lot of insights as to how blood is circulating and what that means in terms of our circulatory system. And then in the last decade, we had people starting to introduce information based on that photoplethysmography, or PPG, to measure blood pressure and glucose. And so it was really the exploratory stages of doing that. So now we're posed, as well as several other groups we understand around the world, are really poised to be able to monitor these 4 types of markers: blood pressure, as well as SpO2, pulse, and blood glucose at the home. And that's what we're talking about in this presentation. So the next slide, I'll go into this a little bit more depth. So just breaking it down, we have 2 different kinds of signals that are really important, and they're both go through quite an extensive amount of understanding by our research team, because we want to get as much information as possible. The pulse and SpO2, that is pretty well known. What you do is, what we do and many others do, is we look at the light absorption and the fluctuations of the light when we have our device measured right through the finger in the red and infrared light, and that reveals to us how much a person is oxygenated, as well as the heartbeat. Blood pressure is more based on what does that shape look like. That shape meaning this wave that goes through as our heart beats. We see this wave, and the waveform gives us lots of information about how fast does that go through, how sharp is the peak of the wave, and what is the characteristic of that wave in terms of how regular is it. So rather than going into all the different terms, at Viit Health, we've learned a lot of things about the dynamics of that system. And that's really important for blood pressure. And it also is important for blood glucose, because in the case of blood glucose, we also add a couple of more technologies. We add polarization and polarimetry, which I'll talk about in a minute, in the next slide. And also the fact that the glucose also trends in several other aspects having to do with the waveform itself. Now, in the regulatory area, pulse and SpO2 have been approved, and many devices are out there, but blood pressure and glucose is still not, it's still at the wellness grade at this point with FDA. So the next slide, let me, and this is just where the current technology is. So what we do at Viit Health, and this is where it gets a little bit more technical, but I think it's useful here to just give you an overview. Our device is shown there on the left in a cartoon form. We're looking at different wavelengths of light going through the finger, and that information is fed into a program that provides files, and our machine learning team uses that. And in machine learning, that's where we use our AI component. In machine learning, what you do is you take all of this information, and we're applying a lot of physics-based analyses based on the dynamics, based on the type of light that we're getting, what wavelength is it, how much is it varying, and then when we use polarimetry, we're using optical filters, so we're actually looking at the polarization state of the light and how that polarization state changes and how the light is scattered. All that information is fed to this workflow, and in the workflow, they take the data, clean the data up, look at the data from different perspectives to pull out features, as they're called, and then train models based on our clinical data and all of these different features to learn what is it that makes certain types of people in certain situations in terms of how we use the technology produce these data, and that provides us with the ability to create a model. Now, one of the things really helping us at Viit Health is the work of one of our engineers who's developed a finger phantom. And we have a fully functional mimic, which is cardiovascular in nature, and it also can have different biomarkers flowing through it. And this is speeding up our ability to create models. So the combination is really important for us. And then as we develop these models, our vision is to have a benchtop device that can do continuous cloud-based monitoring, and that is our predictive platform. So I think this is about it for me, unless there's another slide, I think. Oh, no, one last one. That's right. So just to summarize here, what we do is we have multi-wavelength abilities to measure the light that goes through the finger. So we're able to do two things with that. We're able to note what is the wavelength and how that wavelength is changing over time. So we do what we would call static, or just what is the constant amount of light that's going through, and dynamic and nonlinear dynamics. We combine that with polarization. We use optical filters to get polarized light, and that provides us with a lot of information about what light has been scattered, what is the wavelength, what has not been scattered, and how has that changed based on the amount of blood glucose, for example, circulating in that person's body through the bloodstream. Machine learning is a calibration for individuals. So as the machine learning algorithms get thousands and thousands of patients, we're able now to match the different types of individuals, and that along with some of their anthropomorphic data, that's just sex, height, weight, those kinds of things, help the machine learn and calibrate and provide accurate predictions for those markers that that Victoria showed in the beginning of the presentation. And then our cloud-based monitoring for predictive healthcare, allowing this to be in the cloud, gives us a lot more tools and a lot more ability to do things proactively. So that is one of the key things for us, as compared to just having a device that is only providing information to the patient, so in a static way or just in a real-time way. This is a key feature, we think that is going to be helping us a lot to providing predictive healthcare. The sensor systems are all integrated, which is the key time saving, as well as a very interesting interplay between these parameters. So we've noticed in our work so far, in the clinical work, that there is a subtle interactions between some of these biomarkers, if you will, or some of these vital signs. And that is interesting. So for example, glucose heart rate variability in some people does indicate changes in their glucose, but in others, we have to look at other things other than just heart rate variability. So having a unified platform that has polarimetry, photoplethysmography, and spectroscopy at different wavelengths and real-time dynamics is giving us a lot of that information. So I think that is it for
This
me.
is the glucose. Let me introduce you to the glucose. This is I already said that is the primary source of energy for the humans and mainly for the brain, for the work of the brain. As I said, the glucose after a meal is absorbed by small intestine cells and transported to the circulatory system. In order of this, in consequence, the blood is having glucose concentration around 60 to 100 milligrams per deciliter. Please, the next one. I talked already that if you have low concentrations of glucose, that means hypoglycemia. We talk in medicine, the concentration of glucose in the blood, we call it glycemia, and if it's low, we talk about hypoglycemia, and if it's high, we talk about hyperglycemia. Both of them, hypoglycemia and hyperglycemia, I already said that it can cause disease, but also can cause death. Then it's really important for the body to have constant concentrations of blood glucose. It's for that is so important. Please, the next one. And also, it's for that so important for us. I mean, all the physicians or all the people who are in the health system. For us, it's really important to know the concentration of glucose in blood for the people. Right now, we can do it through different methods, different tests. We can do it at the hospital, obtaining blood from the vein, the venous system, or we can do it at home, for example, using a glucometer that is also taking a small amount, really small amount of blood, but you need blood. And also, already we have this kind of continuous glucose monitoring that they are not taking glucose, but is inserted in your arm, for example. Then, actual glucose testing instruments need a glucose sample. This requires a sharp instrument, which is painful and produce biological waste of challenging disposal, as you can see in this image. As you can see, in all of them, we need blood, and when you're using blood, this is contamination with, as I said, producing waste that is really difficult to dispose of. For that, to have an instrument that can measure glucose without pain, that is great for our patients, and no bloody disposal will be ideal for subject and community. Then, indeed, we have this focus that is great for all my patients. Not only because all the patients who's having diabetes and they need to know his blood glucose sometimes 5 or 6 times a day. Also for people with no diabetes, but he needs to know how is his blood glucose. Please, the next one. We already have more than 800 subjects that we measure with both with venous blood sample to measure glucose, and also, but also we measure blood pressure. We also measure heart rate. We also measure the oxygenation of the blood. Then, as you see, we can have it in this table. We have more than 800 subjects already that we have done all these measures with our Viit instrument and with another standard instruments to measure it. As you can see, this is the data from us, as is common in this kind of research and clinical research. We have a larger number of women that is interested in the study, more than men. This is normal, and we can have here all the data from them. And if we can see the next one. well, something really important in our study is that we have we have people with all the data in glucose from the very low, even lower than 60 milligrams per deciliter. We have a few subjects, but we have subjects with low glucose, and also we have people with high glucose, even so high than 500 milligrams per deciliter. Then we have the complete view of values of glucose in all this study. I'm sorry, in this table we can see, for example, only the subject, because in the more than 800 subjects are from 2 hospitals here in Mexico City. One is the General Hospital and another is Palace Hospital. Here we have only the data from the subjects of the General Hospital. We have more than 600 subjects, and as you can see here, we have the subjects divided in order to understand if some of them are normal glycemic, they have pre-diabetes, or people with diabetes. Most of these people, they don't know that they have, for example, diabetes. We have some patients with diabetes that already are taking medication, but most of them are people who are no diabetes. As you can see, we have the glycosylated hemoglobin, is the way we are divided the subjects. As you can see, if your HbA1c is less than 5.6, you are normal glycemic. If you have between 5.7 and 6.4, you have pre-diabetes, and if you have more or equal or more to 6.5, then you have diabetes. As you see, we have most of the subjects are normal glycemic or pre-diabetic, and we have a fewer subjects with already diabetes. As you see, we have differences between the 3 groups in the concentrations of oxygen and the BMI, and also the neck circumference, for example. Then we have already differences between our population, but that is really good in this study is that most of the people are really happy to know that we are trying to have an instrument that can measure glucose without pain and without bloody disposal. For the subject, without pain, this is really important, and for the community, I mean, for the hospital, for the city, no bloody disposal is really important. Then for this study, we have a very large population that is interested in collaborate with us. Well, that's all. Thank you.
Perfect. Thank you so much. I'd like to move on to the second part of the webinar, which is going to be focusing on some key questions that we've prepared. And I would also like to ask the audience that if you have any quick questions, to please ask them in the polling area. So that's with the buttons that there are below. And so this would be an appropriate time to be asking any questions, and then I can sort through them. If we don't get to your question, we'll definitely message you and be able to answer you directly. So to begin, I actually had a few questions for Tony. My first question was, could you explain in simple terms how Viit Health's technology measures biomarkers using light and AI? Like, what is the secret recipe to using light and AI for this non-invasive technology?
Thank you for the question. So there are a couple of things to mention about the way that we do this interchange. So when we look at light going through our finger, I always like to talk about, you know, the usual issue when you try to take a flashlight and you then go through and put your finger there. You know, on the screen, you can actually see more light than you can see if you looked at it. It all looks red. So that challenge of trying to take that information is something that a lot of other groups have been doing. And even with video, high-speed videography, and just with my face, you can measure pulse, and you can measure even SpO2 or estimate SpO2 pretty easily. So we capitalize on that. but we have a spectrometer inside our device. That is a big difference right there. So I would say that's number one. What does the spectrometer do? The spectrometer separates the wavelengths of light very accurately. So, and our partner that is supplying us this technology is coming up with an even more powerful version of this spectrometer, which will give us even more resolution. Our software is designed to take each wavelength to about 5 nanometers of accuracy. And so now we can separate the wavelength. Now, as our heart beats, we are creating a wave, and that wave goes all the way through. So some people who have the devices where you're looking at bioimpedance, for example, they see that electrical wave that's going through, and they can measure that. And so you could see that as kind of like an EKG, and if anybody has done an EKG, you could see that directly. The wave I'm talking about is the circulation of the blood, and it goes all the way to the finger. So by the time it gets to the finger, there's a little bit of a delay, and in that is the little waveform. It looks like a little mountain and has 2 peaks to it. So that is what we call the PPG. Now, the shape of that wave and how it relates to the second wave. So the waves are cresting, and the waves mimic the pulse. So we get the pulse by just the peak of the waves, but then how those waves are changing over time is another really good clue as to what is going on in a person's circulation. And that impacts things like how the person's responding to the needs of the body. So our autonomic system in our body is regulating things like breathing, regulating heart rate, and all those things. Some of that we have some control over, but most of it we don't, luckily, or else we wouldn't be able to function. So that part of it is giving us clues about blood glucose, and it's also giving important clues about our circulation and our mean arterial pressure as well, those pulses and how far they are. Mean arterial pressure is something I'm very interested in because it's scientifically, it tells us a lot more about what is the pressure in this part of your body, in those capillaries and those veins and those arteries that are right there that we're measuring. The final piece of the puzzle is the polarimetry. And that polarimetry is a special optical technique that we have found a really easy way to do it, a relatively inexpensive but very accurate way because it combines with our spectrometer. So that is giving us information that is key for looking at blood glucose in 2 ways. We look at blood glucose by the autonomic system, the heart rate variability, and the PPG, and this extra method of polarimetry. The machine learning part is more complex, but machine learning is a way of taking all of these bits of millions and millions and millions of bits of information, and based on our technical team, we're telling the machine learning people, these are the things that we think are really important, but their machine learning system will tell us, yes, they do relate to different aspects, and we can use that information to improve our models. So that's a third secret sauce, I guess, that we have, is we have physics-based machine learning, physics-based AI. It's not just AI statistically, but actually based on how we understand how we can measure things with our platform. So hopefully that gives an overview of what makes our technology a little different from the rest.
No, that's perfect. I would like to ask one more question, and then we'll move on to Dr. Adriana. My other question is, what scientific challenges do you face when you're trying to create a truly non-invasive system, and how do you overcome them? So I believe that there are quite a few people who are interested in how we can go into the sphere and space of non-invasive health. And I was wondering if you had any key insights on how to make something truly non-invasive and what maybe the hardships are.
Yeah, the biggest challenge we have is that the body is a dynamic place. So we talk about how accurate is our estimation, how precise are our measurements. The answer is it depends, because the body is changing. So that is a really difficult thing. So la doctora Adriana Monroy is really expert on this aspect, and that's why she talked about this, about blood glucose, is that we have to be very careful when we try to calibrate our system and through machine learning with blood glucose, because it's changing, it's different in the veins, it's different in the arteries, it's different in the capillaries, it's different in the interstitial fluid, in the patch. So that information is challenging. So I would say that's the biggest challenge that we have. So the only way to tackle that is to understand it, work with the body, work with that. So our systems are measuring things in real time for short periods of time, but we understand that the body is changing, and we don't. We want to make sure that our medical group that is also providing data that we're using to build our most precise models are giving us the information, and we are talking to them about what this information means. So anyone who's trying to do this, I think if you miss that step, if you miss the step of the body as a dynamic system, and how do you design a system that works with that dynamism, you have to work with it. You have to get information from the dynamism. And then you have to have the clinical picture very accurately done. If you don't, you run the risk of measuring something that is really just the body giving you a signal, but it's not really the true state of health of a person.
Wonderful, Tony. Thank you. And we've actually been, we received the question. So I'll ask Dr. Adriana Monroy a key question, and then we'll go to the Q&As. So if anyone has any other questions in the audience, please feel free to send in a question and we'll be happy to answer that. So for Adriana, Dr. Adriana, I'd like to ask you, what kind of health conditions do you see this technology addressing first? And what impact do you believe this could have if it was scaled across the healthcare system?
Well, it's very important because the first people I think is going to be really happy to have it is the people who is living with diabetes, because as I said, it's really helpful to have the value of the glucose in real time for the patients, and do it without pain. That makes it easier to have it, right? Because I don't know if you're going to be really happy if you are, functioning your fingers for 5 times a day. In a week, you have 25, 30 fingers. Yeah, that is very painful. But also, it's the fingers that is really sensitive. Then it's really, really bad. Then have a technology that you don't need to obtain a blood sample, that you don't have to use something sharpening that is making pain. That is really great. And maybe people living with diabetes is the people who is going to have to be really, really happy. But I think that it's not only that once. Also, for example, when you are looking for people, because diabetes, for example, is a kind of the disease that you don't have for us pre-diabetes, even some people with diabetes, living with diabetes, that they don't know already, they don't have too much symptoms or signs to alert the people that they have diabetes. When you have, for example, thirsty, or when you are going to urine really often, that means that your blood, your sugar blood is really, really high. Then identify people who is having high sugar blood with not so high values. Then it's easier if you have this kind of technology, because it's not painful. A lot of people, they don't like to go to the hospital or to see if they have high blood glucose, because it's painful. Then obviously they don't like it. They don't want to do it. If you use it in the general population, it's going to be easier to identify that people that is not doing a diagnosis, because for the diagnosis you need a standardized already venous blood sample. But that makes you easier identifying the people who needs this kind of technology, who needs to to use a blood sample, right? Then also in the general population could be really, really good. Not only the people who's living with diabetes, it's also for identifying those people who's having some problems. But also, for example, at the hospital with the health system, for the people in the health system could be really, really good, really easier to identify this kind of people who's having problems with blood glucose, even not only high, also low blood glucose, right? If your people is having hypoglycemia, this is really dangerous, and the people at the hospital identify these people is easier and faster to help them. Then I think all these people is all the society, all I think the all community is going to be so happy, and so this technology is going to be so really, really helpful for all of us, not only for some groups. I think the general population, also because the people who's living with diabetes is increasing really fast in the last years, and it's not going to be, going down. At the time, people with diagnosis of diabetes are going more and more identifying them, right? And also, if you use this, you can identify really, really earlier the problem, not when the people is having a very high glucose problem. Also, if you identify the people in the pre-diabetes, then you can use not only medicine, not only tablets or this kind of medications. You can use diet and exercise, and you can restore the health in those people. Then also, as a preventive feature, is really good.
I definitely agree. I really love the way that you put that, Dr. Adriana, because I think that what you mentioned about the silent diseases that we have, where not these signs and symptoms might not be very obvious, are very much what happens to the majority of us when we hear someone say like, oh, but they seem to be so healthy, right? And I think that instead of relying on our presumptions, and as you said, these barriers to wanting to go and do checkups at hospitals, because of, we even noticed this, and you mentioned this with how many women patients we have actually be part of our protocols versus men. We noticed that some men are kind of have this adverse reaction to blood. And that actually is very important in overall how we can get preventative health to people. So I really love and want to point out that what you said makes a lot of sense in terms of how this could impact the healthcare system to prevent people from having to go to hospitals to do these checkups and actually be able to know their symptoms from before. And I think that there was a really interesting question here that I'd like to ask you as well, Dr. Adriana, and then we'll go into the Q&A as well from other folks that have been asking some questions here. So one of them says, Dr. Adriana, what would you say to other hospitals considering adopting such innovation?
I'm saying that it's great because first, it's not painful. You're not using a lot of material that is very important. For example, to take the blood sample, then you need the sharp, you need alcohol, you need a lot of instrument that also is waste. And I say is difficult, challenging to manipulate this kind of waste, no? Because it's contaminated with blood, and you need to use gloves. There is very difficult. There's more than using this kind of technology is going to be great for the hospitals. Yeah, it's really great because, as I said, for the subject, it's not painful, but also for the hospital is to reduce all this material, right, before the test and after the test. I mean, before the test, because you need all these materials, gloves, alcohol, and cotton, and all these things. And after the test, because you need to manipulate all this bloody waste that is really difficult and challenging to manipulate. And because you have a sharp that is contaminated with blood, this is really difficult. This is really dangerous to manipulate them. I think
Very dangerous.
that in general for the hospital, and not only for the hospital, I mean for the health system, even when you have them in a clinic, in a small clinic, in a rural area, for example, it's going to be great. Yeah, for the big hospitals, but also for the areas, the small areas that you need to check the subjects, the glucose value. And as I said, it's really important. Glucose is really it's just like the other ones are also really important. The heart rate, the blood pressure, etc. But it's more difficult to have the conditions. I think the blood pressure is easier. The heart rate, that you only need like your clock and your hand to measure the heart rate. But for blood glucose, you need, as I said, more technology to measure. And this kind of technology is going to be really, really important and great. And it's going to make it easier to identify the people who is having problems with the blood glucose.
Absolutely agree. I'd love for us to transition into the Q&A section. We have some 12 minutes left in our webinar today, and I think that there's some interesting questions that are open to both of you to respond. And I believe maybe this first one might be addressed a bit more towards you, Tony, where Carlos Trenada asked us, what is the variability of the system by changing its physical conditions? So I'd like to know if there's something that you'd like to mention in terms of how the system varies depending on the different physical conditions in which it can be present in.
So the purpose, I guess, of our clinical trials and the purpose of using our finger phantom is to test it in a variety of different kinds of settings, because we're measuring light going through the finger. So we have differences in scattering, we have differences in the optical path, we have differences in the structure of people, differences in the vascularization of people, and we have noticed that there are gender differences. So male and female, we notice our signals are different. So machine learning is categorizing and compiling, and that's why we have thousands of people, and we're generating millions of data points. And towards the path of regulatory approval, when we've decided exactly what is the indicated use of anything for FDA approval, we're targeting looking at the variability of about 10,000 people or more. So, and with our partners and with our expansion coming up soon, we're on track to do that. It's a great question because that is ultimately the situation. Now, environmental conditions is really important. So there are lots of things that we've noticed, and everyone notices. Some people's blood pressure goes up when they see a white coat, and some people's blood pressure goes down when they see a white coat. Some people's heart rate goes up when they see a white coat. So this is the white coat, meaning the doctor, la doctora there, and her staff. So these are environmental conditions, the conditions that you are taking, the temperature of the room. So we do take those into account. And actually, peripheral skin temperature is something we measure, which is really important. So because it tends to relate to the circulatory system of the person and inflammatory conditions. So this is a very deep question, and there are lots of ways to go down it. And I think we're doing every time we see this, we have one more experience level about the human variability, but certainly in the external variability, we also understand and note these things. And so communication is key for us between machine learning, our technical crew, and our medical crew there that are all collaborating to make sure we understand what all these signals are meaning, especially when we get things that are a little different. So I guess in interest of time, maybe go through a few more.
Yeah, absolutely. No, but I really like your answer, Tony, because I do think that we start to weigh different physical conditions, right? Such as temperature, as you mentioned, right? Like how much that affects the other variables, and there are weighted numbers to each of these. Actually, Dr. Adriana, there's a question for you as well. if you could explain some of the observed relationships you showed between high blood glucose and glycosylated hemoglobin and other physiological variables such as SpO2 or BMI. So I think I can go back to that slide if that makes it easier.
Okay. Well, for example, we have difference, and you can explain, for example, differences between the oxygenation of the blood in between the subjects with different A1C, hemoglobin A1C. Well, as we know, oxygen is captured by the hemoglobin. Then you have hemoglobin with oxygen and without oxygen. We can say this. But also, the A1C is when glucose is tied to the hemoglobin, is joined to the hemoglobin, that modifies the molecule. Yeah, I'm not so sure if this modification is having a lower affinity from the hemoglobin to the oxygen, but that makes difference. That's real. Then maybe this difference we have in oxygenation between the subjects is because we have this glucose in the hemoglobin molecule. Then that makes sense that you have this kind of difference between the different groups, right? Then if you see we have oxygenation lower in the subject with A1C higher. But also can be only because the people is having higher glucose for longer time. I mean, the people that is normal, or people who's having pre-diabetes, or people who's having diabetes, they also have, because of the high glucose for a long time, they have problems with the circulation of the blood. Then maybe also because we're measuring in the fingers, also we can have these differences, not only because the hemoglobin molecule is modified, but also because the circulation of this kind of subject is different between them. Then maybe, as Dr. García said, this is a combination of different factors that is adding this difference. But that is true is that you can see that it's different. The oxygenation in people with diabetes is lower than the people with normal glucose, that has having normal glucose, for example. Body mass index also is different between the different groups because, as I said, when you're having people who is living with diabetes, most of the time is related with obesity. Then, obviously, the body mass index is larger in people with diabetes, and also with pre-diabetes. Even we can see that this is really sad, but the normal glucose group is not having a normal body mass index. The body mass index normal is lower than 25, the value, 25 kilograms per meter, per quadratic meter. Then we don't have a normal value. We have a value around overweighted. Then even people with normal glucose is having overweighted. It's not normal. This is really interesting, but it's normal because our population, this is because the population in Mexico, we have 70% of the population or larger population that is having overweight or are obese. But what you can see is that the people who is larger, I mean, with a body mass index larger, then they have more possibility to be in the high glucose group. Then it's really, really difficult. But the body mass index is not anymore the only way to say that people is having obesity or not, because sometimes, for example, if you have this kind of athletes with a very, very big muscles, then you have a people who is having a body mass index larger even when they are not obese. Then for that, they are using more the adiposity, then the distribution of the adipose tissue as a way to measure obesity. There is really, the amount of people who is athletes or is having a very large muscle is very small in our society, then the difference is really small, but it's better to use another measurings to identify a people who is obese between the people who is normal, right? But one of the very good measurings that correlates with obesity is the neck circumference. It's for that the neck circumference is really important. We have a lot of studies and I show us that the neck circumference is closer to the obesity that is related with a larger adipose tissue, but also a visceral adipose tissue. Not only is adipose tissue, more adipose tissue is where is this adipose tissue, right? Then it's in the center of the body that the neck circumference is really important. Then we can see again that the people who's having a neck circumference larger is because most of the time it's people with obesity that is related with higher values of glucose, and that makes in the group of diabetes, right? And this is the physiological variables why they are different between these groups. Then it makes, as Dr. Garcia said, this is all the physiopathological, this is all the complete view, that is not only one element that makes you have higher glucose value. This is like a complete vision. This is a very complicated vision, because you have a lot of areas that are interacting to have diabetes.
Thank you, Dr. Adriana. I think that that's very important to mention, how all of this data correlates and comes together, right? And how the importance and the significance of how measuring these different groups of normoglycemic, hypoglycemic, and hyperglycemic people will let us see how there are variations in other data points. I think that's fundamentally very, very important to the work that we're doing. I would like to quickly answer 2 of the questions and then finalize our webinar. I would like to mention to anyone who asked the question, we will also be emailing you separately if we didn't get to your question. In terms of Daniela's question, if we've tested on people with varying melanin pigmentation, the answer is yes. We actually take a picture of everyone's hands and we're able to get the specific melanin code. So we're able to have that as a data set as well for each person that we test. For all 3,650 patients, well, and now we have more, we're able to get their specific color, and then that allows us to make sure whether or not there are variations within the system based on melanin. And then for Luis Gomez, you asked whether or not the FDA regular thresholds will be ready in terms of the performance of the device. And we believe that we are already within a lot of the FDA performance thresholds. The only thing that we need to do is go through the FDA pathway. So we are already on set for that with the clinical trials that we will be doing in the US. And in terms of how we see and how we store glucose measurements, we do hope that this will store, and essentially the plan is that it will store everyone's glucose measurements so that you would be able to check in whether you go with Dr. Adriana here in Mexico City or you go with Dr. Tony in Arizona, you would be able to see your evolving measurements of all 5 biomarkers throughout time. So because of how much time we have, I would like to just thank everyone for their time and for attending this webinar. And if you have any separate questions, please make sure to reach out to us. In this QR, you have a link to our campaign, and that's where we'll be posting a lot of information. But please feel free to reach out to us. And in terms of how our timeline and commercialization plan is going and our stage of development, we're more than happy to talk about that with anyone who is interested in this. But the idea is that we will be entering 2026 and 2027 with the idea of commercializing as soon as possible. We're in the last phases of the device and its iteration. So thank you so much, Dr. Adriana Monroy and Dr. Antonio Garcia. It was a fascinating talk to be able to know more about the science and the clinical impact of Viit Health. Thank you very much for your time.
Thank you, Victoria.
Thank you, Victoria.
Thank you to the audience as well.
Bye.
Bye-bye.